when Robin invited me to do this, I said, well, I don't really need an hour and a half because the neurobiomarkers for autism risk don't take an hour and a half. I mean, it could be real short, and I could just say there are none. But I'm going to talk to you about the promise of them, what we're trying to do, and I think some pretty important leads where there is the potential to have a biomarker for autism risk. So as Kathy mentioned, I'm happy to take questions. I don't want to interrupt too much the flow of the presentation, but it's an hour and a half, so we have plenty of time. Particularly, I ask that you interrupt me if there's something that you don't understand, if there's something that I'm saying that isn't clear, if I'm using too much neuroimaging jargon or something like that, just let me know and I'll be happy to clarify for you. So I thought we'd start off with trying to figure out, well, what do we actually mean when we say biomarker? And it turns out there's a lot of different definitions of what a biomarker is, and apparently with the elevated PSA as a molecular blood marker for increased risk of prostate cancer. So we can think about these things in terms of not necessarily a diagnostic biomarker, like temperature would be for fever, but rather biomarkers for risk. You're at increased risk, you should have heightened evaluations, earlier intervention, that sort of thing. So why search for neural biomarkers in ASD? And I'm going to focus today on the neural biomarkers. There are other fields looking into things with metabolomics, with the immune system, looking at other types of biomarkers for ASD, but I'm going to focus on the brain because that's what I study and I know the best. Not that there can't be interaction between, probably the best biomarkers might cover cross-discipline, but I'm focusing on just the brain today. So, as we all know, ASD is behaviorally diagnosed, and some of the things about that I touched on before is that it's quite subjective. You have to have very high standards of getting research, at least in research studies, research reliability on administering the assessments, and it can be a little bit tricky, and it can depend on how the child is doing that day and how the parents report and things like that. I think importantly, there's all of the current evidence that is coming out from the field of neuroscience and autism is that the brain changes seem to be beginning before the behavioral symptoms emerge. So there is some sign of autism that we're not picking up behaviorally that's happening before. And wouldn't it be great to be able to pick that up ahead of time so that we could be right there to identify who's going to have autism and get them into treatment as soon as possible. I mean, as you all know, currently the most effective forms of treatment are behavioral treatments, and the earlier we do it, the better. So in my mind, I think that
using MRI scans to try to figure out who is at this increased risk of having autism is definitely of utility for the field and for families of children with autism. And so we can also think about biomarkers. So there's the earlier detection, earlier intervention. We can think about biomarkers, and I'm going to talk about one in detail today, about predictors of outcome or treatment response too. So not just biomarkers of does the child have autism or not, but can you use a biomarker to say, well, this child has this specific type of autism, and because they have this type of autism, this is what their prognosis would be, this is what treatment they may be able to benefit from the most. So we call this a stratification biomarker. And I think that those two areas I'm going to talk about today, and I think we have some promise there. So first I want to talk, this is the outline. I want to talk a little bit about how we at the Mind Institute use pediatric MRI scanning during natural sleep. So we're doing MRI scans in very, very young children. And the question I get asked most commonly is, how do you do this? How do you get a child through an MRI scan? So I'm going to talk about that a little bit because I think that's relevant if we think about using MRI scans clinically. We want to make it as non-invasive as possible. Then I'm going to spend some time talking about this biomarker of early brain enlargement during the first year of life and how we have evidence that it is a biomarker for a specific subgroup of kids with autism that has some implications for their prognosis. And then finally, I think, I saved the best for last. I'm going to talk about elevated extra-axial fluid, which is a finding that came out of the Mind Institute and actually has been replicated. So replication is the key, I think, in any sort of biomarker study. And that is a finding that we see in 6-month-olds that seems to be predicting autism outcome at 3. So that is, I think, very exciting. So first, let me talk a little bit about how we get an MRI scan and how I think that clinically we might think about moving forward in the field if MRI scans ever were useful in any real meaningful way for helping a child with autism. Right now, it's not. So we typically, if you have a clinical scan up until age 6 or 7 or so, the child will have general anesthesia to have an MRI scan, which is absolutely, the right thing to do if it's clinically indicated for them. But for something like autism, where it's not so clear if there's going to be a clinical significance to their scan, and particularly for research studies, we wanted to take anesthesia and sedation completely out of the equation. So at our imaging center, this is what our MRI scanner looks like before and then after we decorate the scanner. And we actually had, when I first started this study, I had Sally Rogers come over to the imaging center and just give me some thoughts and tips on how she would make this room a little bit more friendly for a child and a family with autism. So we have it decorated like a bedroom, or as close to a bedroom as we can. And we work really closely with the families to scan their kiddos while they're sleeping. So we do our scans at night while they're naturally sleeping. And we send them home sometimes with practice kits, with earplugs and headphones so that they can get some of that sound protection on, work with them at home, and then when they come to the actual imaging center, it just all goes a little bit more smoothly. And we also stay with the kids the entire time and monitor them in case they wake up during the scan. And so I think, over the past 12 years, we've been doing studies using these methods, and it works really well in about 85 to 90% of kids. But it doesn't work in all kids. But 85 to 90% is pretty darn good. And of those, there are a few key things that we can ask that will let us know if it's actually going to be an easy and a quick scan versus one where we have to wait for 5 hours. Because clinically, you're not going to be able to wait for 5 hours to get an MRI scan because the hospital is busy and everything. But there are some kiddos where, say the parent says, well, they can fall asleep in the car and we can carry them in. This is pretty common in young children, whether or not they have autism. Those kids actually are pretty easy to scan, and you can do things like wear them out ahead of time, take them swimming, take them to a jump house, easy things. And then they're much, much more likely to be able to sleep through a scan, particularly, and the younger they are, at 6 months, it's actually quite easy. You just nurse them, swaddle them, and stick them in the scanner. And you can do that without anesthesia. So I think, when I think about, well, why do I use MRI to study autism? Is it going to actually be useful to families? We have to make it in a way that these children don't get sedated for the MRI scans, or as minimally as possible, because they're probably going to need general anesthesia for other things, and we want to not have the exposure be too high. So in the course of this study, so I think I mentioned we have about 400 kids that we've scanned at least one time point during natural sleep. And some of those parents have told me that they've been able to go to their clinical care providers, Kaiser, Sutter, and tell those people, my child can sleep through an MRI scan, and they've actually not had to use general anesthesia for their clinical MRI scans. And that, to me, is making an impact in some way. So that's just a little bit about how we scan these little kids, how MRI might be used as a potential biomarker, particularly in the younger infants. It's a little bit easier. So now I want to get into the biomarker, and I'm calling it a biomarker, a stratification biomarker of early brain overgrowth. But early brain overgrowth in the field of autism goes much beyond a biomarker. This is basically the finding that has been, if you ask anybody about, what's different about the brains of kids with autism, early brain overgrowth is one of the only things and most consistent findings that we've seen. And it actually, I like to point out, it goes all the way back to 1943 in Connor's original paper. He wrote, physically the children were essentially normal, but he noted that 5 had relatively large heads. And that's important too, because even in his sample of 11 children, only 5 of them, a subset of them, were showing early brain overgrowth. And so that's some foreshadowing in what I'm going to talk about here. So the prevailing theory, and when I came into the field with early brain overgrowth, was that all kids with autism had early brain overgrowth. And that this really came from head circumference studies. So this is this graph on the left from one of the most influential papers of the field when I first came into the field around 2005. And this was showing that compared to the CDC norms of what head circumference growth looks like in the first year of life, children with autism seem to be on this accelerated trajectory. The children are born with head circumferences that are not different from other kids at birth, but then somewhere during the first year of life, they go on this accelerated brain overgrowth trajectory. And I'll note that head circumference is a really good proxy for brain volume in the early years of life. And then it's interesting to think about, so head circumference studies started out talking about rates of macrocephaly. And so macrocephaly is a clinical phenomenon where you have very, very large head circumference, and it can be a risk factor for things like hydrocephalus and other things. And in the head circumference studies, people often talked about, this percent of kids had macrocephaly. And because we had norms, we had these CDC norms and other norms, you could define a child as having macrocephaly or not. But then we moved into MRI. And in MRI studies, we didn't have these kinds of norms of, you could say this child has this brain volume, therefore they are, 2 standard deviations above the normal. And so there was a shift when we started studying brain size and in MRI studies to, rather than looking at the rate of something like macrocephaly, or the brain equivalent is something called megalencephaly, a big brain, we shifted to taking group mean averages. And so you just took a sample of children with autism and you averaged their brain volume and you compared it to a sample of children, typically developing children, and their average brain volume. And we did this as well in our early studies, and what the MRI studies have shown is that now on the x-axis ages and years, and what I learned, what we wrote about in this 2008 article, was that early on, before 5 years of life, many of the studies have indicated that there was this 5%, 6% increase in brain volume in children with autism, but that studies of older individuals with autism didn't seem to see these differences. And so what we thought was that early brain overgrowth somehow normalized as people got older. So the thing about the group averages, I think, is important. And then also I'll just comment that all of the data that goes into these slides are cross-sectional studies. So you're studying a different 5-year-old from the 20-year-old that you're studying. They're completely different. And I think that's been something that's been lacking in the field. What we really need to know when we talk about some sort of normalization that might happen is a longitudinal study that follows the same child from age 2 to 20 and to see what their brain growth trajectory is. So we started this project called the Autism Phenome Project at the Mind Institute, and one of the very first things that we wanted to do was replicate this finding in the field about brains being about 6% larger in our children with autism. And so it took about, until we had about 100 kids in our study, was when we started seeing this group mean difference. So here, the children with autism are plotted in red with typical development in blue. And you can see that just like the rest of the field, we see this 6% enlargement when we take the mean of each of these groups and compare them. But take a look at the variability here. And I think it tells a different story. So now we're looking at, in this plot here, is each child with autism. And the black line is just the mean of the typically developing kids. And when I plotted this, I'm like, well, it's not true that every child with autism has a bigger brain volume. Most of these kids fall within one standard deviation of, or one, there are plenty of kids with normal brain size, with brain size that doesn't differ from kids with typical development. But there does seem to be this group of kids up here that do have pretty big brain sizes. And so what we did over time was that we defined a subgroup. We defined a subgroup and we called them this horrible name, disproportionate megalencephaly, which is a mouthful and it's terrible to say. But what it means is that we looked at the child's brain volume and we wanted to look at it in relation to their height, because we wanted kids who had big brains, not just because they were tall and big in general. We wanted kids who had big brains that were disproportionate to their body size. And we pretty arbitrarily took a cutoff. We did a ratio of this brain size to height in all of the kids in our sample. And then we said, let's just focus on the kids who are well above the norm for the typically developing kids. So we looked at the kids who are 1.5 standard deviations above the means for the typically developing kids. So now we're getting away from this, let's just average the kids with autism, compare them to the average of typically developing kids, and we're looking for subgroups. And we defined this group of kids in red here as our ASD with disproportionate megalencephaly. And then you can see that we have plenty of kids in our ASD non-disproportionate megalencephaly or normal brain size group that overlap perfectly well with the kids with typical development. So this idea that every child with autism has a bigger brain is just completely out the window in my mind. And hopefully I can convince you of that too. So then we went, this was when children were 3. So all of these brain volumes were conducted when they were 3. But in our study, we were able to go back and look retrospectively at their head circumferences from their well baby visits from birth. And we wanted to see when does this early brain enlargement happen. And this is the data showing the 3 different groups now, the ASD with the big brains in red, our ASD with normal sized brains in green, and then our typically developing kids. We went and looked at their head circumference measurements from 0 to age 2, and similar to the previous literature, we found that there is no difference in head size among anyone at birth, but only the kids with the big brain, who are going to end up with the big brains at age 3, show this accelerated growth rate at between 0 and 12 months of age. So that's this red line you can see is floating above the others. And that the kids who ended up with the normal brain volumes really didn't follow that pattern at all. So this is just adding to the evidence that this is a subgroup of kids who have this early brain overgrowth. It's about 15% of the kids in our study have this overgrowth. Interestingly, it's happening more in the boys. We actually don't see it very much in girls with autism. And we started to think about this as far as maybe this is a distinct subgroup. They have a different underlying neuropathology. So we've identified this group, at least, of 15% of the boys in our study. So this is only about 21 kids in our sample that had this bigger brain. And then we were like, okay, well, does it mean anything? Are there any clinical implications? Because that's what we really care about. So in the first study where we looked, this was actually kind of a surprise finding. we looked at kids who had a regressive onset to autism versus an early onset. So this, and the field of understanding what regression is and how to measure it has evolved a lot in this time as well. But in general, we think about a regressive onset as a child who has relatively normal or acquisition of skills, but then they regress in some way and lose skills, versus kids who start showing symptoms of autism maybe as early as 12 months of age and seem to be on this path. I had hypothesized that it would be this early onset, the kids who started showing symptoms fairly early on, right about when the brain seemed to be going through this accelerated growth, that would have the early brain enlargement. But of course, as science goes, the finding was exactly opposite. And what we found was that 80% of our kids in the DM group, this big brain group, actually had a regressive onset to their autism. And it was much more evenly split in the kids who didn't have this phenotype. So that was a bit of a surprise, but I think it was a couple things that we thought were promising from this study was that, well, then this early brain overgrowth is preceding their loss of skills later on, because the loss of skills often happens around 18 months of age. And so maybe you could use, particularly our thought was, if you already have a child with autism and you have a younger sibling and you're watching that younger sibling's development very closely, if that child started to show rapid brain growth, their head circumference measurements were getting bigger in the first year of life, maybe that's a red flag to watch that kid more closely for that loss of skills that might be occurring at around 18 or 24 months of age. So I think that was our first indication of a clinical association of this phenotype. And then we've gone on to do another study looking at a maternal antibody-related form of autism. So this is Judy Vanderwater's work, if any of you are familiar with that. She has identified antibodies that are present in the mom during pregnancy that seem to be present in this form only in children who go on to have autism. Children who go on to have typical development aren't exposed to these antibodies in utero. And what we found was that the kids with the big brains tended to be more exposed. There was a higher rate of children exposed to these antibodies than not. And so maybe these antibodies are doing something that's affecting brain growth as early as in utero to affect outcomes later on. The link here between the maternal antibody-related autism and whether a child has regression has sort of come and gone. Again, when you study these subgroups, they get smaller and smaller, so you lose the statistical power to find these differences. So I think in Judy's early studies, the MAR form of autism was related to a regression, but not necessarily replicated. Some of her studies don't have MRI scans in the kids, so we can't tell their brain volumes, but I think there's a story there that we just need to pursue that's linking brain volume and the immune system in some way. So this was all good, and this was all findings that we had at age 3, and it was sort of interesting, and it gave us some indication that maybe we were on to something with this big-brained group, that they did seem, to be have some clinical characteristics and biological characteristics that were different from other kids with autism. But it wasn't until we followed these kids longitudinally that it really made sense to us to think about this biomarker more seriously. So it's a longitudinal study. I'll just put everything up there so you can start looking at it ahead of time. And children enroll between 2 and 3 years of age, and then we follow them at age 5, so 2 years after study entry, we follow them up with another MRI scan and another behavioral assessment. And we're actually currently following this cohort of kids in a middle childhood as well, so 9 to 12 years of age. And what we found when we got to that age 5 time point, because early on I was looking at age 3 to see, well, do these kids have more cognitive problems, cognitive difficulties than the other kids? Do they have higher severity of autism symptoms, something behaviorally that set them apart? And the answer was no, it didn't. But when we looked at them at the time 3 visit at age 5, what we noticed was these kids in autism, they started at a mean IQ of about 55, and the other kids started with a mean IQ of about 65. So lower, but it wasn't statistically different at that point. But then when we followed them out to age 5, we found that the kids with this big brain subgroup were not making the same dramatic increases in their IQ as the other kids with autism. So this is just now plotted the number of IQ points, the change in IQ points from time 1 to time 3, and you can see that the mean for the kids with autism was, or with the big brain group, was 1.5. Most of them centered around not making gains. So up here would be making gains, down here would be losing actually IQ points. Whereas about 30% of kids in the other group were making these dramatic gains, going from IQs with intellectual disability up to normal ranges. And I will note that there are a couple of our kiddos in the DM group that did make dramatic increases. So even within our subgroup, nothing hangs together perfectly. There's probably subgroups within our subgroups or gene interactions, we don't know. Or there's familial macrocephaly, so maybe they just have big brains because their parents had big brains and it doesn't have to do with autism. So I think that this now makes having a big brain, in my mind, sort of an alarm as far as what the prognosis of your child might be. And as we've continued to follow these kiddos out to age 9, they remain the most severely affected in our entire cohort. And I so desperately want to do something to help them. What I can do at this point is continue to try to talk about them as a subgroup and that they need to be thought about differently. So we have looked at the subgroup extensively. I think I talk about them every year here. I do more than this, but I like to talk about them. We've looked at their brains as well. So it's not just that these children have a bigger brain in general. It's not just that every single part of their brain is expanded in the same way. Every single way that we've looked, and I'm not going to go through all of these studies, but we've looked at cortical surface area. We've most recently looked at gyrification, so the folding patterns in the brain. We've also looked at their resting state functional connectivity. So this is now brain activity, measuring how temporally connected distinct regions of the brain are. And we've looked using diffusion-weighted imaging at the structural pathways in the brain. And in every single case, the kids with the bigger brains are different from both the other kids with autism as well as the typically developing controls. They show a different pattern of brain alterations than the rest of the kids with autism. I think the brain is it's working differently. They have poor prognosis. Not to say that there are not kids in this other ASD group that don't have poor prognosis as well, but most of the kids in this group have a poorer prognosis and their brains are different. And so I think it's increasingly I'm becoming convinced that this actually is a real subgroup that is meaningful in some way. So let me go back now to this idea that I told you in the prevailing theory of autism of this normalization of brain volume in adolescence and beyond. So there were those early graphs that I showed you where you had the early brain overgrowth and then less of a difference in older children, in individuals. So this is, again, based on cross-sectional studies. And what I've been telling you is that these kids at age 3 and age 5 are the most severely affected in our group. Well, these kids are not going to be going in and enrolling in an imaging study when they're 12 to 15 years of age because most of them remain nonverbal. They have a lot of problems, and a lot of the studies of older children and adolescents with autism, they don't want people with low IQs. And so actually, usually there's an exclusion of you have to have an IQ of 80 or above to be in our MRI study because you're going to be the easiest type of child to scan. And so these children are not being included in the MRI studies of older individuals with autism. When we look just at their brain, this is now their brain volumes, and again, the red is the big brain group. We've now followed them out to age 5 or 6, and we're following some of them out to age 9 to 12. You can see that there's no sign that their brain volumes are actually going down or normalizing anyway. I think what it is just that they are not included in the studies of older kids, and so you don't see them anymore. So in our longitudinal study where we're following the kids into middle childhood, this was a huge challenge that we faced because when we scanned kiddos when they were little and sleeping, we could scan kids regardless of what their IQ was, it was just how well they slept. But now if we want to follow all of our kids, and I'm very passionate about studying the entire autism spectrum, and There's estimations of 50% of kids have intellectual disability, 50% of kids with autism. We have to study all of them. We have to understand all of them. And so we worked really closely with some really wonderful behavior analysts, actually, to try to be able to scan these kids who are the most severely affected. And this isn't just in our big brain group. This is all of the kids in our study, of the 400 kids. We want to be able to scan all of them and track their brain development over time. And this was a big barrier to getting funding, because the NIH was saying, it's a great idea to do a longitudinal study, but there is no way you'll be able to scan your entire cohort, so we'll end up with the same problem of only studying the people with normal IQs at older ages. And the idea was that you're probably not going to be able to scan a 9 to 12-year-old when they're sleeping, because they're bigger, because their sleep cycles change. And so we were tasked with having to change our methods, get creative again about how we were going to have children who didn't have verbal skills, have low receptive language, and how we're going to get them into the MRI scanner. So this is kind of going back to then this utility of the MRI scans and making sure we can get them. And as MRI researchers, we can have storyboards and things like that, but we're not trained to really work with these children and understand their needs and how to change their behaviors. But who are the ABA therapists. And so we did all these things. We made a rocket ship. We have things like we have a visual timer so the child can be in the scanner. Even if they don't know their numbers, they can see a countdown clock of how long they're going to be in there. That actually helps. YouTube is our best friend. They can watch their favorite videos when they're in there. But the real keys to our success have been enlisting ABA therapists, BCBAs, onto our team, and they work really closely with the children, doing the step-by-step shaping behavior modifications to get these kids, into the building, onto the scanner bed. And it's not easy by any means, and I just want to highlight them because I know that Christina sometimes comes, and Melissa Mello has been really instrumental at the Mind Institute. Audrey Shen actually used to be at the Mind. She's married to Mark Shen, who I'm going to talk about his work later. And that's really where it came about. Mark and I were sitting around, we're like, well, how do we do this? We're so bad at this. And Audrey's like, well, I have some ideas. Oh, well, let's use them. And actually she's now in North Carolina doing this on a national level, trying to get more inclusion of kids with severe challenges into MRI studies. So we do pre-visit consultations. They work with the kids, shaping, pairing, modeling, using the appropriate reinforcements. And it works really well in probably, I haven't done the exact numbers, but 50 to 70% of our kids who are completely nonverbal, who can't even get into the building the first time. We've done things like we have a sensory bin, a cooler of pinto beans with their toys, and we started at the entrance of the imaging center and moved it 5 feet at a time to get a child more comfortable to come into the scanner, and he eventually had a perfectly good MRI scan. So it takes time, it takes patience, it takes these amazing skills of the BCBAs, but it's possible. And I think that's important, and that's something I think that future MRI studies with autism are not going to be able to get away with excluding people with intellectual disability. So I'm just going to summarize this early brain enlargement part of the talk. I hope that I have convinced you that it is something that is useful as a stratification biomarker that could predict outcomes. Not income. In our studies, we've seen that 15% of the boys with autism have a brain volume that's disproportionately large relative to their height. And the clinical significance is they have a higher likelihood of regressive onset, lower language abilities at the time of diagnosis, and then fewer gains in IQ across early childhood. And if anybody is interested in this, I wrote a piece for Spectrum News earlier this year, just going into a little bit more detail about the history of this project and what we've done. But we need to replicate. Replication is everything in a biomarker study, and extend these findings. The current findings are based on a relatively small sample of boys, so it was 15% of boys in our study at the time. So 19 boys and 2 girls make the 21, this subgroup. And then we need to think about developing targeted treatments for this subgroup. So now that we know what that they are making fewer gains in IQ, in thinking about our next grant, we went to Sally Rogers and said, okay, we have this subgroup, they're not responding to treatment. And she said, okay, well, all I can give you is what they've already had. We're not ready yet to do a treatment study in these kids. We need to better understand what their deficits are first so that we can develop treatments for these kids. And then I think importantly, we need to compare them to a group of kids with typical development who have big brains. And that's more relevant for thinking about the cellular mechanisms that might be underlying this brain growth. So just to highlight what we have going on right now, we were recently awarded an Autism Center of Excellence grant. So these are large grants that are awarded. Only 9 are awarded in the country at this time, and this is our third try to get one of these, and we did, basically. And 2 of the projects in this study are focused on this big brain subgroup. One of them is the brain study, which I'm going to plug, because this is a hard group of kids to recruit for. In 6 years of just normal recruitment, we got 19 boys who had this phenotype. If we want to really understand it, we need more than that. And so we're trying to get now 45 kids who have this phenotype. But it's hard because we can't just enroll everybody who has autism. We need to have some indication that their brains are bigger. Fortunately for us, head circumference is a pretty good indicator of brain size, and head circumference is normally taken up to 2 years of age. So we're trying to enrich our sample of kids with autism. We have a whole different battery of passive eye tracking tasks to try to get a better understanding of what their cognitive deficits are. Is it attention? Is it threat regulation? These sorts of things. With the idea that maybe we can tailor an intervention specifically for them to boost their language skills to get them on a better trajectory. But then I think really importantly, we have this third project in collaboration with folks at Stanford called Stem Cells to Brain Cells, where we, from blood samples from the children in our study, we can actually make iPSCs, induced pluripotent stem cells. We can then redifferentiate those stem cells into brain cells and look at the cellular mechanisms of what's leading to this brain overgrowth. How do these brains get larger? Is it because they have more neurons? Is it because the neurons are packed in more tightly? Are there more glia? There are different components that we just don't know why they have bigger brains. And I think that's another therapeutic possibility. If we can figure out what the cellular mechanism is of the brain overgrowth, we could potentially come up with a pharmacological treatment to prevent it or stop it in some way. Let me switch over to the other biomarker that I wanted to talk about today, and that is this elevated extra-axial fluid, cerebrospinal fluid in infants. This is now early extra-axial fluid, so let me just show you what it is. This is an MRI scan, a T2-weighted MRI scan, and normally you would have, can you see my, yeah. Normally the brain fills all of the space, but the white is the cerebrospinal fluid. And in this scan here, you can see that the brain is not filling all of the space, and there is this extra-axial cerebrospinal fluid in these babies that we're seeing. And now the story I want to tell you is to take you on the journey of how we think that this might be a biomarker for autism risk. So this is now looking in not children who are already diagnosed with autism, but using an infant sibling design to look for markers that could be present before the behavioral symptoms emerge. And so probably many of you are familiar with this design. It's something that's used very widely to study early markers of autism. But basically, you follow the early development of younger siblings of kids who are already diagnosed with autism. You start assessing them. I think Sally's early studies, they start around 3 months of age. And the main point of at least the UC Davis infant sibling study was to look for behavioral markers of autism that could be emerging prior to when a clinical diagnosis can be made at 3 years of age. And it's interesting, this study actually came about in 2009 is when we started this. So Sally Ozanoff has been doing this infant sibling study at the Mind Institute for a long time now. But in 2009, Obama came out with the ARA grants, so I don't know if the American Recovery and Reinvestment Act, And Sally said, well, hey, let's just try doing some MRI scans because there's this pot of money. And we applied for the grant. We got a perfect score, which was the first grant I ever wrote, and I got a perfect score. And Sally said, that's never going to happen again in your career. But it was just such a compelling idea. She was already tracking the behavioral development of these kids. Why not just tack on an MRI scan? And we had already shown that we could do scans at night when kids are sleeping. And so we added on an MRI scan every 6 months for a 2-year period during Sally's study. And we basically came up with this finding that's now been replicated. So from infant sibling studies, so the reason why they're effective is that younger siblings have a higher recurrence rate of having autism, so they're about 20% are diagnosed with autism compared to the general population rate of 1%. So when you think about investing resources in scanning lots of kids, and we struggle with this all the time, to find these early biomarkers, you have to, in the general population, you'd have to scan 100 kids, which is a lot of nights at the imaging center, to get one child diagnosed with autism. It's just not feasible. So the infant sibling design was a really attractive way, because at least if we scan 60 kids, we think that maybe 12 of them will have autism. And so that's what we did. And this study was led by Mark Shen, who was a graduate student at the time. And we had 64 infants in our study. 41 were high risk, meaning they had an older sibling with autism. 23 had low risk, and they were imaged at 6-month intervals starting at 6 to 9 months, 6 to 9, 15, 12 to 15, and then 21 to 24, I think. And then they had a diagnostic outcome at 36 months of age. At that point, of our sample of 64, we had 10 who had an autism outcome. So a very, very small study, a preliminary study, but something that I think turned out to be very useful. And one thing that is just how this finding came about was that since we do our scanning at night, there's no MRI technician that wants to be there at night with us, and we can't afford to pay them, so we do our own scans. And what happened was that, this is a pretty typical scan. You see the brain is pretty much filling all of the area, but since we run our own scans, we see all of the scans coming off the scanner, and we started noticing that there were a lot of kids whose scans looked like this, who had more of this fluid surrounding their brain than the other kids. And we're, neuroscientists, we're not clinicians, we're not MDs. When we talk to our radiologist friends, they're like, well, yeah, this is benign extra-axial fluid. This is something that we see pretty commonly in young kids, but it resolves on its own, hence we call it benign. We're like, yeah, but we're seeing it more in our high-risk kids than in our low-risk kids. And I think, this is something that we still need to study, because there are 6-month-olds who have this extra-axial fluid that do go on to be perfectly fine. But within the context of our study, we were seeing it more often in our high-risk infants, and they were indeed getting called by the radiologist more frequently with this, benign extra-axial fluid, or there's lots of different ways that they can call it, and not every radiologist calls it the same way. And so being neuroscientists, we said, okay, well, qualitative, understandings are good, but we need to quantify it. Let's measure it. Let's measure it and analyze it. And so we developed ways, and it's not really that difficult, but one thing I'll just tell you with MRI labs is what we usually do is we'll get an image, and the first thing we do is strip off the skull. So you can see here that this is the skull. And normally if you're not sitting there at the scanner acquiring the images, you might just feed it through your automated pipeline and strip off the skull. And if you did that, you would never see this elevated extra-axial fluid, right? Because once you take off the skull, you can't really tell that there was extra fluid there. So this was just sort of a fortunate, I think, byproduct. Yeah? Are
they sleeping?
Yes.
So when we sleep, you want to fill in the blanks of when
Go
we
ahead.
sleep?
I don't want to presume I know what you're going to say.
So there has to be controls with children that you don't think would be autistic, because when we sleep, our brain actually shrinks. The cerebral spinal fluid circulates,
Yeah.
and that's how basically
clear out all
the toxins that
Yep.
we're accumulating during the day. So
Yeah, So you're right that it's sleeping, and that is a finding that has. So we quantified it, and we waited for the kids to grow up, and what we found was that the children who went on, the 10 kids who went on to have an autism diagnosis are in red here. There's a group of children who have not autism but other developmental delays. They're in green. And then our high-risk kids, so kids who had an older sibling with autism but ended up as typically developing, are in blue, and purple is, in purple and blue is our low-risk kids. And you can see that across all of the time points from 6 to 24 months, the kids with autism had elevated levels of this extra-axial fluid. And so we were really excited about this as a potential finding at 6 months of age. And it's interesting that the group with developmental delays other than autism also show this slight elevation early on, but then it does resolve, and they have more normal levels by 24 months of age. And but what was really striking was that these guys were high when we first looked at them, and they remained high throughout the time period that we were evaluating them. So this is all great and everything, but it's 10 kids. It's 10 kids with autism that we studied here. So what we did was we contacted the IBIS Network, which is a large-scale, it's an Autism Center of Excellence network study, where they now have multiple sites, lots more funding than we had. And they have the same design, same longitudinal scanning, and they have 343 kids in the study. So Mark actually went on to do his postdoc with Chapel Hill with Joe Piven, who is the PI of the IBIS study. So we were able to replicate or attempt to replicate these findings in a much larger sample, which is really, really rare and exciting to do in autism science. So he went on to Chapel Hill, where they had a much larger data set. Now they had they had a sample of 343 kids, 49 of whom ended up with autism. Most of them did not. The high-risk negative was 193, and then the low-risk negative was 132. So a much larger sample, a much more respectable sample. And they found the exact same thing, which again, never happens. And this was a group where, it's a multi-site study. They were getting their brain scans. They were really interested in brain overgrowth. But the first thing they did was strip off the skull. So they never saw this extra-axial fluid, never thought to quantify it. But when you look at the scans with the skulls, they had babies that looked just like the ones in our study with this extra space in between the brain and the skull. And indeed, what they found in their 49 kids who ended up with autism was a very similar pattern of elevated 18% elevation of extra-axial fluid in the dorsal part of the brain that persisted across all 3 time points. And as an extension of the original findings, because they had so many more kids in their study, they were able to look at a group of kids who had more severe symptoms of autism versus a group of kids who, I think they used ADOS severity scores, less severe. So the more severe kids actually had even greater amounts of extra-axial fluid than the less severe kids, so the less as rated on the ADOS. So this isn't looking at IQ or anything, it's just looking at autism severity. So now going into this whole biomarker idea again, what a lot of the biomarker field likes to use is pattern classification. What is the accuracy of using this type of method to predict who's going to have autism, which I think is useful in some ways, but I don't think it's everything, and I worry a little bit because that's what the field is so focused on. Here we saw that the amount of extra-axial fluid that a child had at 6 months of age had a 70% accuracy in predicting autism. So that means that, based on some threshold that we picked, we could accurately say this child will have autism in 70% of the kids. But that means 30% of the kids we couldn't. So this, I think, as a clinical utility is not that good. But what I think you can use is is an indication of risk and an indication of looking more into the pathogenesis of now we know that extra-axial fluid is doing something in a lot of the kids, 70% of the kids, but whether we can scan every child at 6 months and say you're going to have autism or not, I don't think we're there, and I don't think that's how we should think about using this as a biomarker. I think maybe in combination with other factors like brain size or other things, we can get those accuracies more reliable and more replicable, but we're not there yet. So I'm not trying to sell the point at all that extra-axial fluid, that every 6-month-old should get a scan and that will tell you if they're going to have autism. But I think it's interesting to think about in terms of risk, and particularly in terms of the infant sibling. These kids, if I had a 6-month-old and an older child with autism and they showed up with extra-axial fluid, I would certainly be looking more closely. But interestingly, I will say, as far as the replication go, in the IBIS sample, they had enough kids to do this pattern classification, and they came up with the accuracy of 70%. We then went back and applied that same classification algorithm to our UC Davis sample and got the same accuracy rate. So I think we're measuring something real here. We just have to figure out what it's telling us. So this was all in infants, so this was 6 months to 12 months of age, and in this infant sibling design. So these are people who already probably have some sort of genetic disposition to autism. We wanted to look at extra-axial fluid in what we call a normal risk cohort, so where there is not necessarily a multiplex level of autism, because lots of people have just one child with autism. And so we did this by looking in our Autism Phenome Project cohort of 3-year-olds. So now they're already diagnosed with autism, but we have a mix of kids who have either multiplex or simplex autism in that group. And we wanted to look to see, is extra-axial fluid also a marker, could it be a marker for autism in a simplex, in a normal risk cohort? And indeed, we did find that the kids with autism are plotted here in red. They, even at age 3, have elevated levels of extra-axial fluid. There's this group of typically developing kids who also have extra-axial fluid, and I'd be very curious to follow up with them to see, if they ended up having other types of problems, learning disabilities, things like that. But this is where we found that the group of kids with elevated extra-axial fluid, so the kids at this threshold, which is the same threshold that we used in both of the infant sibling studies. We used this threshold of the ratio of CSF to brain volume of 0.14. When we looked at the kids above that threshold, they had significantly more sleep problems than the rest of the kids. So this is getting back to the idea of what is CSF actually doing? How is it related to sleep? If your sleep is disrupted, could that be doing something bad, not only to your mood or your parents' mood, but to actually your brain development as well? So I think that extra-axial fluid, just to summarize, I think it's a promising biomarker for autism risk. I think it's important because it's observable and it's reliable. So going back to our definition of what a biomarker is, it's something that you can see. We could see it when we were just scanning the kids. It's not something that you have to take back to the lab and measure and use really fancy processing methods. You can just see it and measure it. It may also inform our mechanism. We now have ways that we want to look into the mechanism of disease. We have not seen this in animals at all, so this could be because it would be great. This is like the perfect thing for an animal model where we can do more invasive types of research. So David Amaral also works very heavily with primates, non-human primates, and they do developmental studies, and we have never seen extra-axial fluid in a primate or a rat or a mouse. So it could be something specific to humans. It makes our job a lot harder, I think, because it'd be a lot easier to take the CSF out of an animal than a human. But it's detectable with any structural MRI scan. These are MRI scans that you can do in like 2 or 3 minutes, so it doesn't take long. And just to point again that it's been replicated like in the exact same way, which never happens. So we do still have more work to do. We don't yet know if it's specific to autism. We have that indication that other neurodevelopmental disorders, remember that green group of kids who had developmental problems, not autism, had more elevated extra-axial fluid. Mark is working with Fragile X, so we see it in Fragile X as well. So it may not be specific to autism, but I think it's something important that we should think about in terms of neurodevelopmental disorders. We've also been trying to think about how we can do an epidemiological study of this, to look at the prevalence and look at how extra-axial fluid, now that we think that there's something more than, that it's actually not completely benign, we could look for people who've had MRI scans with larger numbers in the general population and see who ends up having autism. So like Kaiser would be perfect for this, but we haven't been able to get in the door there, to look at how prevalent is this in the general population. So, any questions on the extra-axial fluid part? I'm just about finished here, so we can open up to a more general discussion as well. Did you have a question?
Yeah, and I don't think there's an answer yet, but the question that comes to my mind is this more likely to be cause or effect? So is this a compensatory mechanism if we're thinking about the CSF as having
Sure.
a clearing effect? Might there be more need for it in people who have something metabolically going wrong that the body is trying to compensate for?
Yeah, no, exactly. I mean, and you're right, we don't have the answer to that, but it does open all of these doors to exploring mechanisms that we didn't think about before. You know, very recently there has been this renewed interest in what is CSF. You know, when I was in school not that long ago, it was just the shock absorber of the brain. There really wasn't much. But we've discovered the glymphatic system. We have these sleep discoveries. CSF is way more important than what we previously thought. So we need to look into how it's related in autism and other neurodevelopmental disorders as well. Was there another question?
So first regarding your big brain study, I wanted to say thank you for focusing on this issue of inclusivity in the autism research world. We think about inclusion and equity a lot with regard to autism versus non-autistic kids, but not so much within the autistic world.
Exactly.
And so I appreciate that focus in your research.
Sure.
The question I have is whether or not you suspect there's any crossover between these two biomarkers that you're looking at. Do you think there's a shared mechanism here, or do you think that these are really two distinct biomarkers that are highlighting two distinct mechanisms?
So it's a great question. I didn't talk about it because we don't really know what to make of it, but in our infant studies, both in the UC Davis and the North Carolina study, the kids who had extra-axial fluid, that higher extra-axial fluid, also had bigger brains. Not necessarily in that, in that threshold of 1 point, in that disproportionate megalencephaly subgroup of really, really high brains, but they had bigger brains than average, and so there probably is a link there, but we haven't, in our big brain group, we don't have 6-month-old scans to know what their extra-axial fluid looked like. Although I will say that some of them in our big brain group do have extra-axial fluid, and they still have it at 9 years of age, so probably they had it at 6 months, although we don't have the scans to prove it. But we just need to look at the interaction better within all of this. And as far as inclusivity, I'll just say that my other line of research is into sex differences in autism, and I am also extremely passionate about including more girls in our research study. So send your girls our way, too. That's a different study. It's called GAIN, and I talked about it last year. But I'm all about studying everybody who has autism because they deserve it. So I'm just going to end now with, I want to give these studies their due credit, because there are several studies that have come out in the past year using these pattern classification strategies, and I mention them also because they tend to get picked up in the press, and so maybe some of you have also read about these studies. 2 of the studies, the Hazlitt and the Emerson papers, were using the IBIS cohort, so the same cohort that we were using, and they got published in very high-profile journals, basically saying that they could differentiate kids with a 96% accuracy using resting state functional connectivity at 6 months of age. This sounds really great and it's really promising, but I just want to caution that these studies are not replicated yet, and that oftentimes in these pattern classification type studies, you can train an algorithm to classify your sample of kids. You go through all of these iterations and permutations, but how well that generalizes to a completely independent sample of kids is completely unknown. And I am very risk averse, but I would bet money that you would not be able to replicate a 96% accuracy rate in another sample. I don't know, like maybe $5, I don't know, but maybe a little bit more. But I think these studies are important, but I think there's more to the field of biomarkers in autism than just finding this magic diagnostic marker. I hope that I've convinced you that we can use MRI in other ways other than saying we can scan you at 6 months and predict if you're going to have autism, because I think that it's misguided to think that MRI is going to be able to do that completely, and so I don't want to lead to a lot of disappointment in the field, but I think that there are other useful ways that we can use MRI. So the third study was by Bill Boesel and Chuck Nelson at BU, so it was a different sample, but they used EEG at 3 months of age to predict autism with a 90% accuracy. And these just seem just so good. And the other problem with these is that these are deep learning algorithms that don't actually tell you what that algorithm was using to classify the two kids. You can't ever figure that out. So how useful is that really? Whereas with something like extra-axial fluid, we started with the finding and then we went out and looked at how predictive it was. But we can still go back, and even if it's a 70% accuracy, we can go back and study lots of other things. And so I would just caution when you see and these tend to make the big headlines, but we actually don't know what the algorithm is looking at. So, yeah. Oh, you need the microphone. And I promise I'm almost done. Sorry.
I had a question. As a clinical provider, I see lots of kids with autism, some of whom have big heads. Are we at the point when we can actually tell families about this being a risk for poorer prognosis, especially since we don't have tailored treatments?
Yeah.
Or should we even talk about it? Because this is terrible news for a family to hear.
I know. I struggle with that too. I would say that it's important for them to know what our studies have shown, but without overpromising from the results of our studies. Because, and now, I find that we're recruiting, so actually send them our way, because we're recruiting to try to study this more. We have this sort of preliminary finding. We need to see if it's real. And if it is real, if it does really predict a poorer prognosis, that will be useful. But there were those several kids in our big brain group who did really well. So when I talk to families now, I say, we just don't know enough to have a complete prediction, but we're working on it. It's not great, but I wouldn't go out and ring any alarm bells for sure.
Hi, Christine. That was a great talk, and it's really exciting findings. I have just a couple ideas about treatment, because, of course, we've done a lot with Fragile X syndrome, and they have big heads and increased extra-axial fluid. And what's going on molecularly with the low levels of the Fragile X protein, that mTOR pathway and the mGluR5 pathway, which leads to a lot of proteins that accelerate replication. We have started a targeted treatment study looking at metformin, which lowers that mTOR pathway. Yeah. And actually metformin is also protective against cancer because it lowers these proteins that are important for replication and enhanced cell cycling. Mm-hmm. And that's, of course, occurring in Fragile X, and we've seen a great response to metformin even in young kids, ages 2 and older. Right. So we should talk more about maybe looking at what metformin does to that enhanced brain growth and extra-axial fluid, and would that be a treatment for another subgroup other than Fragile X syndrome? Yeah. And you can use different molecular biomarkers like MMP9, which is important for brain connectivity, which is way too high and interferes with brain connectivity when the FMRP protein is low. Mm-hmm. And actually, FMRP protein is supposed to be low in the brains of individuals with autism without Fragile X syndrome. So this may be very important for that subgroup. And then there's mTOR downstream markers that you can look at. But the other treatment that comes to mind is bumetanide, which
Mention this,
is
yeah.
this diuretic, okay, that helps with the GABA transition from stimulatory to inhibitory. And I haven't read that Bozell et al. Paper, but maybe that has to do with GABA deficits or delayed transition in this GABA exchange. But bumetanide is a diuretic, supposed to be good in autism, also thought to be good in Fragile X, and maybe that diuretic would help to get rid of some of that extra-axial fluid. So that's another possibility for treatment. And I don't know if the Ben-Ari studies are looking at, you know, this subgroup of big brains and extra-axial fluid, but they're doing big studies in Europe. And, you know, we need to think of some of those studies, you know, here in the U.S.
Yeah, no, I completely agree. And I think that those treatments would be really exciting for the big brain, at least with our stem cell approach of you know, growing brains in addition. We can test pharmacological treatments before doing
you can
it in children.
sprinkle these meds on the stem cell
Squirt 'em on.
neuronal cultures.
Yeah.
My husband laughs at me when I say sprinkle these meds on, but actually it's a great way to
Well,
look at
you
how
know,
neurons communicate in culture
Absolutely.
from iPSC-derived neurons and how these neurons communicate with the sprinkling on of some of these new meds.
Absolutely. So we'll get to you.
My question is about the basics of the study. You, as well as the speakers this morning, talked about using the Mullen to measure IQ on these kiddos. Are you using the visual reception subtest, or are you using the whole instrument? Because the whole instrument would confound language and motor skills too.
We do use the whole instrument, and we can look at the subscales separately. We can look at verbal versus nonverbal skills here. In our study, it was across the board. It wasn't specific to any of the subscales in the group, and we also used it to group language. I mean, the reality is it's tough to study 3-year-olds with or without autism, and finding the right instruments it's hard, but we have to deal with what's available to us. So recognizing the limitations, I think, is
My
important.
practice is primarily those kiddos under the age of 3, and I was wondering if you had something more that I didn't already have.
Yeah, no.
Thank you very much.
I don't.
Okay, we're going to do one more question.
Well, he
Actually, it's not a question.
Oh.
It's a comment. I'm wanting to warn against your concern about the deep algorithm. Reproducibility is everything, but in the world of technology we have now, we can apply deep algorithms anywhere, essentially. And artificial intelligence is expanding so quickly that we are going to live in a world that has a plethora of meaningful and useful deep algorithms that we will probably never appreciate the foundations of. So I wouldn't be too cautious about deep algorithms. I'd be very cautious until they're reproduced.
Sure. And we're using machine learning in our lab to do these things, so I think it's worthwhile to pursue, and I think the technology is only getting better. So thank you for that. So I'm actually finished. Like, I just had a summary and then acknowledgement slides. So I like to just show off our MRI team, because it's been a lot of hours where we get to know each other, and so we have a lot of fun at our Mind Institute annual thank you party, because we have all these hours at the Imaging Center to dream up these themes and costumes that we can do for the party. So a lot of work has gone into discovering these things, but I think it's been worth it. And then we have this, as I said, this new Autism Center of Excellence grant where we're following up on the big brain kids, and it's all very exciting, and it's all happening at the Mind. So that's it.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies upcoming events, and how you can help make a difference.